Hi everyone. So today I'll be talking about the topic immune cell mediated drug and gene delivery and its application in immunotherapy. Well, first of all, I would like to give a brief introduction about the immune system. Uh, the immune system is a sophisticated biological defense mechanism which comprises an intricate network of cellular and molecular components, and this system encompasses specialized cells including lymphocyte, macrophage, then dendritic cells, which collaborate each other to identify and neutralize the potential threat. And at its core, the immune system operates through two integrated branches: the innate and the adaptive responses. The key organs such as thymus, thymus and thymus, spleen, and lymph nodes. They serve as critical hubs for immune cell production and activation. Well, the bone marrow, a primary lymphoid organ that generates and matures various immune cells, while the thymus they educate the T lymphocytes and the secondary lymphoid tissues, including the spleen and the lymph nodes. They facilitate the antigen presentation and initiate an adaptive immune response. Well, these immune systems, they have like remarkable capacity to distinguish between the self and the non-self entities, including that enables the precise targeting of pathogens while minimizing the collateral damage. And through the complex signaling cascades and cellular interactions, this biological surveillance system constantly patrols the body, maintaining homeostasis and protecting against a cascade of potential health threats. And then they have their own ability to interact. For example, these immune cells, they interact with the tumor microenvironment, and this tumor microenvironment, they represent a complex ecosystem characterized by the interaction between the diverse immune cell population and the cancer cells, all or also known as neoplastic cells. Within this extracellular space, the innate immune cells such as macrophage, mast cells, and neutrophils, they engage in dynamic crosstalk with the adaptive immune cells, and they primarily, for example, the primary one is the lymphocytes. And these cellular interactions, they are mediated through an elaborate network of signaling molecules, including chemokines, adaptive cytokines, as you can see here, they are releasing. And this molecular dialogue, they facilitate a delicate balance between the pro-tumorigenic and anti-tumorigenic responses, and that shape the tumor progression as well as metastasis. And the plasticity of immune cells within this tumor microenvironment, they allow for their functional reprogramming, leading to the emergence of distinct phenotypes that can either promote or inhibit tumor growth. Therefore, understanding these interaction is crucial for developing the targeted immunotherapeutic strategies, which can aim at modulating the tumor microenvironment to enhance the anti-tumor effect. Next is we'll talk about the different kind of cancer immune response, and that can have interventional therapy that can be effective for interventional therapy and its the various delivery modalities. Well, the intricate interplay between the cancer and the immune system, they represent a complex landscape for therapeutic interventions within the tumor microenvironment. A diverse array of immune cells, including the T lymphocyte, as I said earlier, the natural killer cells, and antigen presenting cells, they engage in dynamic interactions with the cancer cells. And the interventional therapy such as transarterial chemo immobilization, and then ablation and internal radiation, they have emerged as a potent modulator of this immune extracellular space. And these approaches not only directly target the tumor cells but also induce immunogenic cell death in releasing the tumor associated antigens and the damage associated molecular pattern that can stimulate systemic immune responses. And the efficacy of these interventions is further enhanced through innovative delivery modalities, including the nanoparticle based system and there is advanced delivery methods that facilitate the precise targeting of these therapeutic agents to the tumor cells, and that optimize uh, the local concentrations while minimizing the systemic toxicity. So, in addition, the combination of the interventional therapies with immunomodulatory agents, for example, immune checkpoint inhibitors, they have shown a promising effectiveness in amplifying the tumor immune responses, and they overcome the immune suppressive mechanism within the tumor microenvironments. Well, within the pathological states, the immune system, they mobilize a sophisticated cascadal process of cellular defense to combat the invading pathogens and the aberrant cells, and the neutrophils and macrophages, such as they act as a frontline responders, and they employ phagocytosis to engulf and destroy the foreign entities. And these natural killer cells, they exhibit the cytotoxicity activity against the virus infected or even the cancer cells, any kind of foreign particles. While CD8 positive T cells, they directly eliminate the compromised cells through the targeted cytolysis, and CD4 positive helper T cells, they facilitate the immune response by activating and modulating other immune cells. And the B lymphocytes, they produce highly specific antibodies that neutralize the pathogens and mark them for destruction. And the dendritic cells, that is also one of the immune cells which serve as a crucial antigen presenters, and that breeds an innate and adaptive immunity by activating the T cells. And the innate lymphoid cells, they secrete the cytokines to regulate the inflammation and tissue repair process. And the regulatory T cells, they maintain immune homeostasis by suppressing excessive responses, while memory T and B cells, they provide the rapid and enhanced protection upon the subsequent pathogens that encounters, and forming a basis of immunological memory. Well, in the immune cell mediated drug and gene delivery, which represents again a bio-inspired approach that harness the innate capabilities of immune cells to act as Trojan horse for targeting therapeutic delivery. And this strategy, they exploits the natural migratory and homing abilities of various immune cell population, including the macrophage, neutrophils, and T cells, to target uh, the biological barriers and accumulate at the site of disease by loading these cells with therapeutic cargoes. And it has been found that they can overcome the traditionally impermeable obstacles such as blood-brain barrier, and that enables their access to previously unreachable targets. And the intrinsic tropism of immune cells, they have like tropism towards that target area, and the place which has been affected by through which like, for example, inflammation or through the infection or any neoplastic facilitates the precise localization of these therapeutics and minimize the off-target effects and reducing the systemic toxicity. Furthermore, this approach capitalizes on the extended circulatory half-life of the immune cells, and that provides again a sustained release platform that prolongs drug exposure at the target site. Well, in the context of homing and migratory mechanism in the targeted drug delivery, they have a landscape that encompass a facet array of homing and migratory mechanism, and each they exploit unique biological phenomena. The enhanced permeability and retention effect that leverages the aberrant vasculature of the tumors, allowing passive accumulation of the nanoparticles in the malignant tissues. And the active targeting strategies, for example, surface modified nanoparticles, or they encompass with the they add on with the ligands, and that enables the specific receptor mediated uh, interactions with the target cells. And that for one of another example is the exosomes. They exhibit intrinsic tropism at their surface protein that facilitates the natural homing to the particular site where this cancer cells are present in the tumor microenvironment. And the stimuli responsive nanoparticles, they harness the tumor microenvironment's distinctive characteristics such as acidic pH or elevated enzyme levels to trigger the localized drug release. And that cell penetrating peptides, when they are incorporated into the nanoparticle design, and They even help in enhancing the cellular uptake and intracellular delivery by facilitating the membrane translocation. So these diverse approaches, they are synergistically contribute to the evolving paradigm of this precision medicine, offering a tailored solution for targeted therapeutic interventions across various pathological conditions. Again, talking about different methodologies for drug loading in the advanced delivery system that encompass again a diverse set of approaches. For example, ex vivo modification techniques involving the manipulation of the cells or even tissue outside the body to incorporate the therapeutic agents. And in case of in vivo targeting strategy, explore we can even exploit the endogenous cellular machinery or even the mechanism or the molecular interaction to achieve site-specific drug accumulation. Again, in case of nanoparticle, various scientists have even shows that approaches like encapsulation and delivery, and the passive loading methods such as thin film hydration or even solvent evaporation facilitate drug incorporation during the nanoparticle or liposome formation. And in case of active loading techniques, for example, post formation strategies like pH gradient or even electroporation or even solvent exchange to enhance the drug loading efficiency. And also the endogenous loading technique, they harness the cellular machinery to package drugs into the exosome, while surface conjugation employ the covalent chemistry. There are many different kind of approaches again to attach drugs directly to the nanoparticle surfaces. And the stimuli responsive loading mechanism is also used that integrate the again environmental sensitivity that allows for triggered drug release in response to specific physiological uh, cargoes. And these diverse loading strategies again they collectively contribute to the development of highly customized and efficient drug delivery system that enables the precise spatiotemporal control over the therapeutic agents that distribute agents distribution and release. Again, there are various applications of this drug and gene delivery through these immune cells. In the context of application, one of the famous example is CAR T therapy, and that represents a personalized medicine which harness the power of adaptive immune system to target malignancy with unprecedented specificity. And this innovative approach, it involves the ex vivo genetic modification of the autologous T lymphocytes to express chimeric antigen receptors which is engineered fusion protein combining the antigen recognition domain with the T cell activation motif. And upon re-infusion, these reprogrammed cells exhibit enhanced tumor recognition capabilities and amplified cytotoxic potential that enables them to overcome immune evasion mechanism, which can be employed by the cancer cells. While initially they are developed for the hematological malignancies, the ongoing research is expanding CAR T cell application to solid tumors and even to infectious diseases and inflammatory disorders. And the versatility of this platform extends to even gene therapy, where CAR T cells that serves as a vehicle for targeted delivery of the therapeutic gene, and that offers even potential solutions for the genetic disorders and expand that expands to the frontiers of regenerative medicines. Well, again, in the context of challenges, again, if we talk about the pharmaceutical development, manufacturing, the scalability that presents even multifaceted challenges, which it can include even the process optimization, equipment design, and quality control across increasing production volume. Concurrently, increasing the production volumes, the off-target effects, that also is a big problem because it's one of the hurdle in drug efficacy and even safety profile, and that necessitates the advanced pharmacological strategies to enhance the specificity and to minimize the unintended biological interactions. In addition, the regulatory considerations further complicate this landscape, demanding the rigorous documentation, validation protocol, and the adherence to evolving global standards. And these interconnected factors collectively influence the trajectory from bench to bedside, requiring a delicate balance between the innovation, safety, and compliance to navigate the complex path of drug development and commercialization. Now again, there are several emerging technologies in drug delivery which are revolutionizing this field. In case of therapeutic approaches through the innovative strategy, for example, biomimetic nanoparticles, they are engineered to mimic natural cellular structures and offer enhanced biocompatibility and targeted delivery capabilities. And these nanoparticles, they exploit again the indigenous cellular mechanisms to overcome biological barrier and improve the pharmacokinetics. And in addition to that, the delivery of CRISPR-Cas9 systems presents a paradigm shift in gene editing therapeutics that necessitates again the sophisticated vectors to facilitate intracellular transport of ribonucleoprotein or even complex while maintaining their integrity and functionality. And the lipid nanoparticles, the engineered viral vectors, they are at the forefront of this technique, and that enables again precise genomic modification in vivo. Further, the combination of these therapies, they leverage the synergistic drug interaction and multimodal diverse systems which are emerging as a potent strategies to combat complex diseases, and that again offers a potential for enhanced efficacy and reduced drug resistance through simultaneous targeting of the multiple different pathways.